This talk is about what I've learned over the last 40-odd years, and how I would like to see the next generation of biological anthropologists think and teach critically about evolution, although possibly with updated cultural references. I want to start off with a sort of foundational catfight in evolutionary theory nearly half a century ago. It was precipitated by a 1980 paper by Stephen Jay Gould called "Is a New and General Theory of Evolution Emerging?" and the response by Ledyard Stebbins and Francisco Ayala. No, it's not. A central issue for them was whether the reductive evolutionary theory stemming from population genetics was in fact adequate to explain paleontology. Gould argued persuasively that some things, like mass extinctions, had huge impacts on the history of life on Earth that could not be extrapolated from population genetics. Now, before I go any further, full disclosure. Stebbins and Ayala, a few years later, both wrote me letters for my first faculty job. Nevertheless, it was also clear that just within our genetics department at Cal Davis, there was a lot more going on in the genome than simply point mutations which may or may not turn out to be good for you. In 1980, there were hardly any university programs in organismal biology. We nevertheless knew that in one end of the black box goes molecular novelty, followed by mutational degradation over generations. And out the other end comes new bodies interacting harmoniously with their environment. The human genome sequence at the end of the century brought us no closer to knowing how to make a four-dimensional human body from a one-dimensional string of DNA bases, much less how to make two different bodies from two different sets of DNA instructions. Moreover, there is more to speciation than simply the accumulation of mutational difference, because somehow species are composed of potential mates and competitors for mates, and new species prefer to socialize and mate with their own kind. But how they figure out who is in their own kind is still an open question. We now know that animals don't passively occupy pre-existing niches in an ecosystem, but are involved in actively creating the niches that they occupy. And learning is a major part of the survival strategy of many animals, especially mammals, and cultural evolution, broadly defined, has certainly been a major part of our evolutionary history in addition to our biology over the last 3 million years. And although Michel Foucault rarely enters into this conversation, certainly the intervening decades have demonstrated the interpenetrations of evolution and politics, recurrently packaged to rationalize social and economic hierarchies as having evolved naturalistically for the optimal benefit of all. But ultimately, whether these are add-ons to or differences from the earlier generation's evolutionary theory is kind of a stupid question. What is clearer now than it was back in 1980 is that there is a lot more to evolution than simply deviations from Hardy-Weinberg equilibrium. And especially when it comes to humans, the science doesn't operate in a cultural vacuum. In fact, history shows that the people who yell the loudest for the separation of facts and values are invariably the ones bringing the cringiest values to their facts. For a notable example, the reason that The Origin of Species is still readable today is that Darwin avoided talking about people. 12 years later, in The Descent of Man, he talks about people, and there is something to cringe at on nearly every page. Notably, Darwin, for his time and place, was a progressive when it came to racism, but the situation is a little bit different when it comes to sexism. Darwin tells us that there is a difference in the inherent mental powers of the two sexes, and he's not going to try and prove it, he's going to take it for granted, and just try to explain it naturalistically. And he knows that his position is not the progressive one. With respect to differences of this nature, mental powers, between man and woman, it is probable that sexual selection has played a very important part. I'm aware that some writers doubt whether there is any inherent difference, but And you just want to say, please stop right there, Charles. Don't complete that thought. But he does, because he is not one of those who thinks that men and women have equal intellectual capabilities. And then a little bit further down the page, that's how man became superior to woman. Ta-da! And don't think that this is inconsequential. In the 1920s, the creationist and early feminist William Jennings Bryan will cite this passage to prove how fucked up Darwin was. Now, I might add that even telling you about this is risky, because there are elements within the evolutionary biology community to whom raising any sort of criticism of Darwin is effectively violating a religious taboo, like depicting the Prophet Muhammad. But here's my point. A century ago, Bronislaw Malinowski wrote in Nature that evolution has been mishandled more frequently than any other principle, and he called out specifically Herbert Spencer and Ernst Haeckel. That's the frontispiece of Haeckel's 1868 book, which was translated into English a decade later, but without this charming picture. 100 years later, why do we have to repeat Malinowski? Why is evolution still being mishandled more frequently than any other principle? I'll tell you why. Because it's about kinship, which is a symbolically powerful subject, and it is science, which is a source of cultural authority. So it involves most fundamentally myth-making for the modern world. But by now we've seen it all. We know what the bad stuff looks like, and we know it's going to keep coming. So how can we do it better? Or more precisely, how can we think about and do biological anthropology anthropologically? What would an anthropologically informed biological anthropology look like? Well, the first thing we have to do would be to reject the Enlightenment holdover idea that the incorporation of cultural, political, self-interested values are exceptional pathologies in science that need to be ignored or dismissed. We now realize that they need to be studied and analyzed, for we are all products of our time and place, and we wear cultural filters that shape the way we see everything, including evolution. The best we can do is consciously make those cultural filters as transparent and as benign as possible. Over the course of the 20th century, the scientists who were interested in telling more culturally inflected histories of science generally self-identified as Marxist scientists, rather than as amateur cultural historians who had recently discovered political economy. The geneticist J.B.S. Haldane was high on the list, and in an article for Labor Monthly in 1941, he explained how the Hegelian negation of the negation, achieving synthesis by juxtaposing opposites, was a good way to understand biology. Well, it's not, and I don't even know if it's a good way to understand intellectual history. Nevertheless, if I may riff for just a second on the idea of negating the negation, negation itself implies a pre-existing binary state, thesis, antithesis, and that is actually what requires negating. The idea that complex things in human life come in binary oppositional states. Some simple things do. Some organic molecules do, notably enantiomers. Claude Lévi-Strauss insightfully highlighted binary oppositions in human myths, and I think there is indeed powerful and cross-culturally recognizable symbolism in binary oppositions, like right and wrong, good and evil. But in human biology, that is a straitjacketed way to think. The problem is that not much actually comes in nature as binary opposites requiring a synthesis. Rather, nature comes as a spectrum whose endpoints are culturally opposed to one another. You can juxtapose night against day if you like, but in so doing, you lose the Earth's rotation that makes night and day actually transient snapshots over a 24-hour period. That is to say, in the complexity of human biology, cultural binaries are far more common than biological binaries. Thinking about biology in a non-dichotomous fashion renders it generally more accurate, and the contrast of extremes is generally uninformative and inapplicable for the majority of people. So I would like to see us recycle the classic negation of the negation in Haldane's attempt at a progressive biology as just a broader rejection of dichotomous thought, negating the idea of negation itself. The second idea comes from the work of two very non-Marxist scientists, Sewell Wright and Theodosius Dobzhansky. Wright flirted early on with the American Eugenics Society, but stayed mostly apolitical after that. Dobzhansky, of course, took the lead in anti-eugenics, anti-Nazi, and anti-racist biology, and they were long-term collaborators on the meaning of genetic diversity in populations of fruit flies, and presumably beyond. This was very much in reaction to the biology of the eugenicists, which imagined genetic uniformity as optimal for a population. Wright and Dobzhansky's population genetics began with genetic diversity, or polymorphism, and heterozygosity as a benefit to the organism and to the population, and especially to the future of the population. And the idea of unity in heterogeneity rather than in homogeneity is a progressive one that sounds even better in Latin on your money. Third on the list is what Haldane called the transformation of quantity into quality, but is more generally the recognition of levels and hierarchies in biology. In one sense, this is really the foundation of Darwinism. You get little changes in breeds, varieties, subspecies, and eventually it translates into bigger changes in different genera and families. The problem is that the changes don't just sum together. Hierarchies and higher order organization in evolution were promoted by the molecular evolutionist Alan Wilson, whose classic paper with Mary Claire King came out my first semester in graduate school and certainly shaped my initial interest in these questions. And by the mid-1980s, it was clear that first, there was no simple relationship between how much mutational difference existed between two animals and how different they appeared to be phenotypically. The same amount of mutational difference between humans and apes couldn't predict how different two fruit flies at the same genetic difference would seem to be from one another phenotypically, or how different two frogs at the same genetic distance from one another would seem to be. And second, that point mutations are a small part of what's going on in the genome. Genetic difference maps onto phenotypic difference to a first approximation, but it clearly takes more than the mere summation of nucleotide substitutions to make a new species or genus. Things in biology are systems and are rarely additive. Fourth, in 1871, Edward Tyler's concluding thought in Primitive Culture, the first English text of anthropology, was that the science of culture is essentially a reformer's science. He meant that in a specific context, in which primitive peoples were being exterminated on account of their ostensibly primitive biology. Tyler's argument is that their biologies are not inferior, their cultures are. That's an argument for ethnocentrism and against extirpation, which is a step in the right direction. He is literally freeing up indigenous peoples from the tyranny of genocidal biology. But from within biology itself, I point to John Beckwith, who co-led a group in the 1960s that worked out the lactose operon in E. coli, the first system of genetic regulation. And rather than brag about it at their press conference, they talked instead about concern for the dangers of genetic research. And in the immortal words of their colleague, the noted racist and sexist James Watson, seeming more ashamed than pleased with their neat science, James Shapiro and Jonathan Beckwith of Harvard Medical School held with much fanfare a press conference to announce their new way to isolate specific genes was on the pathway to eugenically motivated genetic engineering of human beings. Knowing of their left-wing views, I, like most of my colleagues in the Boston region, saw their self-denunciations as manifestations of unrepentant leftist fears that further genetic research would render inviable the communist dogma that assigned all social inequalities to capitalistic selfishness. And I am so pissed that I didn't take an Ussie when I met John Beckwith. In that same press conference in 1969, Beckwith criticized the slumlord activities of their employer in Cambridge. This was before Richard Lewontin got to Harvard, but I think Lewontin managed to put the class interests of science on the table in a way that had not previously been noted. He was an especially powerful critic of first-generation sociobiology, as it explored social hierarchies naturalistically and segued smoothly into rationalizing human hierarchies. Moreover, Lewontin was especially critical of the sociobiologist's sole reliance on competition as the creative force in the evolution of social behavior at the expense of cooperation. I only met him once, and he told me he didn't like the way I spelled my last name. True story. Steve Gould, I think, was instrumental in calling attention to the social politics that has accompanied and influenced science, especially evolutionary biology, over the course of its history. He said where he came from politically and how politics and evolution are entwined, and he challenged others to stand up for the politics entwined in their own science or to disown it. And in retrospect, the political positions and alliances of, say, Matt Ridley, Richard Dawkins, and E.O. Wilson don't look very admirable under scrutiny. Wilson was friendly with the racist psychologist Philippe Rushton. Viscount Ridley is an apologist for oil and gas. And perhaps Gould would have been as hateful as Richard Dawkins if he had lived longer, but somehow I doubt it. He signed my copy of Ontogeny and Phylogeny, May the creationists never have a day of peace. The last idea I want to put out there actually forms a nice segue to the rest of this hour. It's one that we associate with the geneticist Conrad Hal Waddington from the University of Edinburgh, who was highly respected in the genetics community, but was regarded suspiciously within the evolutionary biology community. Why? Because he wasn't a reductionist. Instead of asking what a gene does and how many alleles there were in a population, he asked, where do phenotypes come from? And the answer he found is that phenotypes are stimulated by the environment, and you can select for the amount of environmental stimulation it takes to produce the phenotype. And eventually he was able to mimic Lamarckian evolution within a perfectly Mendelian and Darwinian framework. This was threatening within normative mid-century evolutionary biology, which was resolutely anti-Lamarckian. At any rate, in this context, Waddington had been influenced while at Cambridge by the philosopher Alfred North Whitehead, who was developing an anti-reductive metaphysics of biology. Waddington's lifelong best friend from college was Gregory Bateson, who went into the softer science of anthropology and eventually became Margaret Mead's third husband, after Luther Cressman and Rio Fortune. Whenever he visited New York later in life, the geneticist Waddington stayed with Mead. Mead and Bateson produced an anthropologist, Mary Catherine Bateson, and Waddington produced an anthropologist, Dame Carolyn Humphrey. This is just my way of saying there's a lot of anthropology around Waddington and Whitehead, which may explain why I've enjoyed diving into this rabbit hole. In brief, what I can extract from Whitehead's Metaphysics of Biology is this. 19th century Darwinism involved understanding things through time, as Isaac Newton had established the general laws of motion for physics. But now, says Whitehead, we're in the 20th century, and we have a different understanding of the physics of things. We know that things aren't distinct from time. We know everything exists in space-time. Newtonian physics is three-dimensional. Einsteinian physics is four-dimensional. And if we want to base our biology on our knowledge of the physical world, then we should be thinking in something beyond Darwinian terms. Now, of course, that's easier said than done. If you agree with the proposition that our representations of biology should be predicated on our conceptions of physics, then perhaps a Newtonian Darwinian world is an obsolete one. But what might replace it? Whitehead believes that matter in motion is a three-dimensional view of nature. Rather than concentrating on three-dimensional things as our building blocks, he encourages us to look instead at four-dimensional processes as our foundation. What we perceive as a thing is here the intersection of different processes, and this intersection is a temporarily stable state that we perceive and register as a thing. And the reason that a thing may have surprisingly nebulous boundaries and be elusive to define satisfactorily is that it is a convergence of four-dimensional processes being squeezed into three. Of course, I'm not the first person to be playing with these ideas. My own ideas have been inflected by the philosopher John Dupre and by the cultural anthropologists like Tim Ingold and Gisli Paulsen, who have been influenced by Whitehead's ideas. The basic starting point is, first of all, that relationships will be privileged over things, which I think should be unexceptional to anyone who's worked in social anthropology. After all, social relationships are not located within brains, they are located between brains. They just have to be extracted from the mouths and actions emanating from within brains. While there may indeed be a correlation between brain size and social group size, it's not at all clear how social group size plugs into human kinship and social relations, which vary culturally, unlike human brain size. And second, that becoming will be privileged over being, which again should be unexceptional to anyone who's been working in evo-devo over the last few decades. And here the reductive and mechanical model of the universe has been eminently successful since the Enlightenment, but it has also led us to idealize a species or even ourselves as frozen in time at the midpoint of reproductive age, without primary concern for how we got there or where we go next. On the right is the famous image accompanying Pioneer 10 out of the solar system, in which NASA engineers sent into space a picture of what they wish they looked like after a bikini wax. What we would like to do to a second approximation would be to think historically rather than mechanistically. Rather than think about transformations of state through time, let's foreground the processes that are converging to produce the things and states we perceive, not the least of which are the production of idealized views of body form. More fundamentally, to be a functioning human adult, you not only have to have the proper physiology, but you also have to know how to walk and talk, which aren't instinctive except to the extent that there has to be a template available to instinctively learn it from. More importantly, you need to have learned right from wrong and to choose right, or else you're just not going to last long anywhere. You've had familial life experiences in ethnic households rooted in traditions of your ancestors and your social class. You've had individual experiences that have shaped your personality, perhaps traumatically, and continue to haunt your dreams. You're motivated on account of obligations you have incurred and ambitions you strive for. Those are the processes ultimately making you become what you are and what you will be. You are formed by the intersection of those processes. The idea then is that the mechanical model of things at different points in time is reducing a four-dimensional tesseract into a three-dimensional cube. A tesseract doesn't exist in three dimensions. It exists in four dimensions, and all you can ever get in three dimensions is a freeze frame. And that is literally all I know about tesseracts outside of the Marvel Comics universe. But to return to biology and processes, in the 20th century it became anathema to talk in any sense that sounded possibly teleological, with classic papers parsing the term to try and let good goal-directedness in and get bad goal-directedness out. But the fact remains that a fertilized egg is heading somewhere, namely to senility and death. Sorry for the pessimism, but that's the way our life history evolved to be, although passing through happy, vigorous, fertile adulthood on the way. You can see this as a ball of cells doing some things, and granted that's all it is, just cells, but it's the coordinated activity of the cells and their relationships that evolve to function towards the goal of gastrulating, neurulating, becoming an embryo, and eventually a tadpole. It's this issue of working with 4D processes in 3 dimensions that is the root of the fact that central biological concepts are often rigorously definable only at the introductory level. What once seemed like bounded 3D objects turn out to have fuzzy boundaries and states of quasi-existence the closer you look at them. Here's a stretch of about 10 or 12,000 bases of DNA near the tip of the short arm of human chromosome 16. And in green there you have 2 unambiguous and actually very simple genes, both of which have the same DNA coding sequence and make the exact same protein, namely half of hemoglobin, the alpha half, transporting gases in the red blood cells of your circulatory system. So there are definitely 2 genes here, alpha 2 and alpha 1 by anyone's counting. The question is, are there any others? At the right, in blue, is a stretch of DNA with a base sequence about 80% identical to that of alpha 1. We know that it's transcriptionally active, but we don't know when in development or in what tissues it works, or what it actually makes, aside from the fact that we can predict that it looks a lot like alpha hemoglobin. We also know that its presumptive coding sequence, when compared across species, has a considerable excess of synonymous mutations, which is a hoity-toity way of saying that it's evolving like a functional gene. Is it a gene? I'm going to say yes, and move on to the DNA sequence at the left, which is also about 75% identical to that of alpha 1, base for base, but is not transcriptionally active. We call it pseudo-alpha 1. So it's structurally similar to a functional gene, but not itself a functional gene. It's called a pseudogene because it's structurally a recognizable gene DNA sequence, but not functionally a gene. Even more genetically ambiguous, here are 5 allorepeats, or short interspersed elements, each only about 300 bases long. They're the products of RNA transcripts from a source somewhere in the genome that was reverse transcribed into DNA and reinserted into the human genome at various times and in various places, the copies themselves not being functional. So once again, it's an identifiable structural element of the genome, but not itself functional. So are we looking at genes here? And if so, how many? Definitely 2, probably 3, possibly 4, arguably 9. And if we look at the fine structure of the gene itself, it's in a box because it's a thing and things have edges. The 2 intervening sequences in alpha globin itself aren't very interesting, but introns can contain all kinds of weird genomic elements within them. But more importantly, the box begins at the beginning of RNA transcription and ends at the nucleotide that gets a poly-A tail added to the messenger RNA as part of its processing. So we can arbitrarily define the boundaries of the gene as being the boundaries of the slightly processed messenger RNA transcript, which is perfectly sensible. But that leaves out the promoters in front of the gene and the enhancers in back of the gene, without which that gene would be nearly, if not fully, as dead as that pseudogene. And of course, there are all kinds of associated biomolecules attached to the DNA sequences that regulate it, package it, unwind it, and extract its information. If you regard the DNA as a one-dimensional instructional sequence, then you ignore the actual physicality of the gene itself, which does seem arbitrary and reductive. Moreover, this DNA sequence is valueless to the bloodstream if you don't have a functional beta globin also, making the other half of hemoglobin on a different chromosome. And it's just my way of pointing out the difficulty in precisely defining, identifying, and delimiting the gene at the molecular level. Now, of course, this is not news. The gene is many things to many scientists. But if it is essentially amorphous and has no edges, then how is it really even a thing? Actually, the very first thing a gene ever was, from the fruit fly work of the Thomas Hunt Morgan lab, was a difference maker. It was a name for the basis of how and why one fly was different from another, and they were arrayed on a chromosome like beads on a string. In the 1940s, bacterial geneticists began to realize that those differences were manifested at the cellular level as different proteins, and that genes were themselves proteins. In the 1950s, they realized that no, the genes were the DNA that was making the proteins that were making the differences. And in the 1960s, they realized that not all RNA gets translated into proteins. So there were classes of RNA differences that did not manifest as protein differences. The introduction of information theory permitted geneticists to regard the gene as a unit of information, transmitted faithfully across cell generations by mitosis and across organismal generations by meiosis, and this was high school biology by the 1970s. Nevertheless, the invention of DNA sequencing showed a whole lot of things in the human genome that have some, but not all, of the features that were being attributed to genes in the genome. Things like pseudogenes, viruses, long and short interspersed elements, satellite DNA. At the same time, the sociobiologists following Richard Dawkins were talking about genes as replicators, even if replication sites and information sites in the DNA don't map onto one another. Some of you may even remember young Dawkins rhapsodizing about genes. They are in all of us. They created us, body and mind, and their preservation is a main reason for our existence. They have come a long way, those replicators. Now they go by the name of genes, and we are their survival machines. The genetic determinists, among whom Dawkins became something of a figurehead, and who of course predated the gene itself, nevertheless latched onto the gene as the cause of their economic, political, and military superiority very early on. One of my favorite quotations is in fact from R.C. Punnett, whose eponymous square is still memorized by freshman biology students. He wrote the first college textbook in English on Mendelism, called Mendelism. And here is its last line, the thought he wanted students to take away from learning about the gene. Permanent progress is a question of breeding rather than of pedagogics, a matter of gametes, not of training. As our knowledge of heredity clears, and the mists of superstition are dispelled, there grows upon us, with an ever-increasing and relentless force, the conviction that the creature is not made, but born. No. But to return to reality, or at least to biological materialism, we now know that a single gene can be responsible for several different RNA and protein products, and that the causal chain from DNA sequence to phenotype is a lot more complicated than it once looked in bacteria. So where does that leave us? If a gene is all, or even some of these things simultaneously, and you can't even tell where it begins and ends, then why even bother calling or regarding it as a thing? It is a node, an intersection of various cellular, physiological, and intellectual processes. Rather than asking what a gene is, we can instead look at the cellular, physiological, and intellectual processes that the gene is involved in. Obviously, it's involved in whatever the cell it inhabits is doing, and it is a product of DNA duplications that have left some functional and some non-functional copies of the DNA sequence. It's involved in the production of bodies, as well as the transfer of cellular information across cell and organismal generations. Moreover, it's a connection to the ancestors, as well as a legal argument for leniency in sentencing. The poor gene is doing a lot of work. Now, this has all been said before, but I'm using the gene as an example of where it might be profitable to take a processual approach to making sense of what we have traditionally seen as the elemental things of genetics. Another central thing in human biology is the organism. Once you get outside of our species, problems with the idea of individual organisms arise quickly, like fungi and ant colonies. But even with humans, the more biology, the more complexity. Maybe my thingness is established by my consciousness and memories. I think, and therefore I am. But if I can't remember anything before the age of 2, does that mean that's when I began? I'm glad I remember my childhood and not someone else's, because that would be weird. But weird changes of consciousness do happen. That's why people believe in spirit possession. Moreover, my consciousness was shaped by the things that have happened to me. Does that make the circumstances of my life literally part of me? There is genetic continuity with my earlier self, but because of cell division and cell death, there aren't actually any cells in me now that were alive in me 70 years ago. There are descendant cells. So what makes me the same person I was 70 years ago is to some extent cellular continuity. But once you go cellular, you run into the continuity of the germplasm, August Weismann's 19th century term for our cellular descent from all of the generations before us, which is way too much for the present purpose. And clearly there's a difference between a human being and a 200-pound vat of human cells. Moreover, you are cellularly incomplete, and likely dead, without your symbiotic microbiome. And finally, you are constantly shedding your epidermal cells, which is why they can get your DNA off of pretty much anything you touch now. So from a cellular perspective, you leave bits of you everywhere you go. Let's parse this with someone we can all agree was not just an organism, but a damn fine organism. Elvis Presley. So what do we mean by Elvis? Do we mean the thing that started when he was born and ended when he died? That's a useful legal definition, essentially from when he began breathing air to when he stopped breathing air, which is generally around the same time as one becomes brain dead. But obviously it excludes the 9 months that he was inside his mother's womb gestating. Was that embryo not Elvis? It certainly was genetically or cellularly. But obviously Elvis was more than his cells, and more than his cells plus the bacteria and other microscopic life forms that ultimately comprise a human body. Much of Elvis was made by Colonel Tom Parker, although not genetically. And even talking about Elvis in the past tense presupposes that there is no longer Elvis, for he has passed on, kicked the bucket, shuffled off this mortal coil, run down the curtain, and joined the choir invisible. And yet Elvis isn't fossilized. He is physically still decayed flesh and bones. There's still DNA in his cells. His cells may not be happy, but they've still got their DNA, from which you could, in theory, clone Elvis. And a quarter of that DNA is in the cells of his 3 grandchildren. When NAGPRA was passed in 1990, it opened up the question, not just for Native Americans, but for everybody. What is the relationship between personhood and the body? Does personhood expire when you die? And if not, then what is the relationship between living Elvis and the bones in his grave? Does he live within them? And what is the relationship between Elvis and his grandchildren? Does he live within them? Or does Elvis live within all of us as the embodiment of certain things and ideas in mid-20th century America evoked by his image? But what actually was Elvis? Is Elvis alive? Rather than thinking about Elvis as a 3-dimensional organism composed of cells, who started breathing, got fatter and drug addicted, and eventually stopped breathing, let's try and think about Elvis instead as an intersection of processes whose convergence produced what we see as the organism who was born in 1935 and died in 1977. Some of the processes were cellular and genetic, some were circumstantial, some were social. He was a body that occupied space and changed through time and is still doing so. He was, as Richard Dawkins said, a vessel for the replication of genes, and they replicated very successfully in his trillions of cells. But his body had and needed other cells in addition to his own. And of course, Elvis is not alive physically, but is he still alive genetically and culturally? And if we can get anthropological for just a minute, it is important to remember that any specific human organism is also occupying a slot in a network of familial social relations. In the context of Game of Thrones, people can be representatives of lineages, which can consequently be considered realer than the organisms temporarily occupying the slots within them. At any rate, here is my first crack at identifying a human organism as the temporarily stable intersection of a set of processes. There is, of course, the stuff that comes from your parents, which includes but is not limited to your chromosomes and mitochondrial DNA. Obviously, the previous generation passes on to you a lot more than just your DNA. There are also the processes by which a zygote gestates, begins breathing, eating, speaking, locomoting, eventually maturing, and senescing. There are the processes that allow us to survive by creating a fit between the body and a specific environmental variable. Long-term and irreversible, like the adaptations to hypoxia. Short-term and reversible, like tanning. Adapting genetically, physiologically, and of course culturally. There are the processes by which we acquire the knowledge it takes to survive, and in humans that period is particularly lengthy and active. Again, Tim Ingold has written on this subject. There are processes of socializing and interacting with other organisms, competing, cooperating, and just living. And then, of course, there is the golden rule of doing unto the next generation that which was done unto you. In humans, that has something to do with genetics, but a lot to do with technology and culture more generally. That, in addition to his rotting corpse, is where I think we can still find Elvis. And I want to wind down with something that is confusing in its classical sense, namely primate taxonomy. If we ask a simple question like, how many extant species of primates are there? It turns out that that number has undergone a remarkable transformation in the last few decades. If you asked that question 40 years ago, you got a number around 170. If you ask that question today, you get a number well over 500. This would, on the face of it, seem to indicate that the primates are doing remarkably well, speciating like mad and being remarkably prolific. That is, of course, nonsense. Well, what accounts for the multiplication of species among the primates in the last few decades? Hypothesis number 1. They're not going extinct at all, but are actually doing very well and proliferating like mad in the wild. That is false. They really are going extinct. Hypothesis number 2. Maybe a whole bunch of new species have been discovered in the wild interior of Africa or on Kong Island. Again, false. In the last few decades, there have been some previously unencountered species discovered, mostly lemurs on Madagascar, but certainly not hundreds. To understand what has happened to primate taxonomy, we need to understand that the primates are endangered, and that primatologists are in a very different position than astronomers are. If you're in graduate school and you're interested in Jupiter, you can be pretty confident that Jupiter will still be there when you finish your degree. But if you're in graduate school and you're interested in mountain gorillas, you don't have that confidence. Consequently, the number one priority of primatology is to save its subject matter. A few years ago, if you published an article in the International Journal of Primatology, it would be accompanied by a little cartoon of an aye holding up a sign saying, "Let us live," in Latin. I don't believe the editor was trying to speak directly to the Catholic Church, but the point is that primatology begins as advocacy for primate conservation in the modern world. So to understand the recent history of primate taxonomy, we need to understand the hard work that primatologists have been doing with international governments to draft legislation to protect the primates from loggers and miners and plantations. And these laws are generally written at the species level. This species needs to be protected. Well, if you're a logging company or a mining company or a plantation owner, you might do a study of the distribution of the primate in question. And then you go to the courts and say, this species of primates lives in part of the forest A and in part of the forest B. We just want to log or mine or plant on part of the forest A, but you'll still have that primate species in part of the forest B. So let us clear half the forest. To which the primatologists respond, actually, the species in Forest A is a different species from the primate in Forest B. Your information is out of date, and both of those species are now twice as endangered as that one was before. So you can't log or mine or plant there. Now, before I go any further, let me make sure everyone understands that most primates are very highly endangered. What I'm describing here is strategic, but just one weapon in a dwindling arsenal. At any rate, in this way, primates are saved, at least in theory, and what gets sacrificed is just what we are calling a species. It's not that the number of primate species is less accurate than it used to be, or that the number of primate species is more accurate than it used to be. It's that we're now tabulating different things. What we were calling a species 40 years ago was along the lines of a unit of population ecology. What we're calling a species today is more along the lines of a unit of conservation. And every primatologist agrees that saving the primates is more important than what you call them. Once again, like gene or organism, there is an obvious and simple definition of what a species is, that we can teach in introductory classes, and that we owe to the great ornithologist Ernst Mayr, the biological species concept, in which a species is officially groups of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. Since speciation itself is a process, not an event, or at least a process to geneticists and an event to paleontologists, we have all kinds of intermediate states in living animal groups. It sort of became the official species concept for evolutionary biology in the 20th century, in spite of the fact that, as Ledyard Stebbins pointed out, doesn't really hold for plants, so it's consequently zoocentric. And as George Gaylord Simpson pointed out, it really can't be applied to the fossil record. Consequently, Simpson focused rather on the unique evolutionary niche and lineage that a species represented, and simply avowed that it was operationally equivalent to Mayer's definition. In other words, a species was a thing to both scholars, and you just took their word that they were denoting the same thing. At any rate, neither of them was considering the influence of cultural issues, like conservation politics, in defining what a species is. So maybe what a species is, once again, is a 3D question in a 4D world. Maybe a more productive question would be to ask, what does a species do? In other words, what work is accomplished by trying to identify the units of the living environment? First of all, every species has a niche. It used to be thought that niches came prepackaged in the environment and species simply expanded to fill them. But now we see adaptation as a more active and creative engagement between species and environments. And thanks to natural selection, everything eventually gets recycled. Everything consumes and is in turn consumed by the ecosystem. May be ultimately consumed by wolves or by worms, but bodies don't last forever. They are nevertheless food for other species. Second, a species is a reproductive community in which participants can either potentially mate or compete for mates. They are able to recognize conspecifics and to interact with those conspecifics appropriately. Third, a species is a label for us to linguistically identify and distinguish animals and plants and impose some kind of basic order upon them. That's a very human thing to do. And there is often a broad convergence between the species recognized by indigenous peoples and the species recognized by the people studying the indigenous peoples. Higher order categories, however, tend to be more incompatible as they are based on local criteria for grouping. Fourth, in the world of industrial deforestation, a species has a new role in facilitating political action to save the animals. And of course, you name a species and the biology community will know your name forever. What then is a species? Well, whatever the heck it is, it's doing a lot of things as a temporarily stable state at the intersection of evolution, capitalism, conservation science, scientific reputation, and governmental action. As we've noted, it obviously has something to do with the gene pool and a niche to which it is adapted at any point in time. It also has something to do with systems by which animals identify others like them or not like them in crucial, that is to say, reproductive ways. And they are also constructs of the mind and how we decide that this is a kind of that and not something else. And they are units of political praxis for environmentally minded activists who are using the weapons at their disposal for warding off the encroachments of greedy corporate capitalism. And finally, we tend to think careerism plays a bigger part in the processes of making a paleoanthropological species. But if we want to talk about what a species is at any point in time, it is a convergence of these processes. Anyway, if I were still teaching, I think this is the way I'd be teaching about species. More generally, of course, this is a first tentative intellectual step towards making sense of biological anthropology in the 21st century anthropologically. I've taken bits that I think are interesting and relevant from diverse scholars, and I'm certainly not prepared to get into an argument with anyone about Whitehead and his ideas. It's just a possible place where I'd like to see the textbooks of the future go. And so to conclude, here is a bit of a reading list for deeper reflection.